Polypeptide synthesis. What is this process and how does it work? To put it simply, there are two key steps to polypeptide synthesis: transcription and translation. Transcription is the process of making an mRNA copy of a DNA sequence. This mRNA molecule is then used to assemble amino acids, forming polypeptide chains, which creates the second part of the process known as translation. Transcription begins in the nucleus, specifically the DNA, or deoxyribonucleic acid. DNA is made up of molecules called nucleotides. These nucleotides are attached together to form two long strands that spiral to create a structure called a double helix. A nucleotide is made up of three parts: deoxyribose sugar, a phosphate, and a nitrogenous base. Along the DNA strand are genes, containing the instructions for making proteins. When a gene is switched on, the enzyme RNA polymerase attaches itself to the start of the gene, moving along the DNA, creating a strand of messenger RNA, or mRNA, out of 3 bases on the non-coding strand, reading the strand from the 3' to the 5'. The non-coding strand determines the order of which 3 bases are added to the mRNA. The mRNA bases are always complementary to the DNA bases. Cytosine will always pair with guanine on the mRNA strand. Guanine will pair with cytosine. Thymine will pair with adenine. However, the adenine will pair with uracil on the mRNA strand, as the mRNA does not have thymine. All T bases have been replaced by uracil or a U base. In this model, we will be using the following DNA sequence, which is found on the non-coding strand. The RNA polymerase starts with the T base, and so will start the mRNA strand with the complementary A base. The RNA polymerase continues to do this, building up the mRNA base by base as it moves along the gene. To carry this genetic information out of the nucleus, mRNA carries a transcribed copy of the relevant instructions from the nucleus to the ribosomes in the cytoplasm. It is small enough to move through the nuclear membrane via nuclear pores, as it is only a single gene long. The original DNA strand never exits the nucleus, as the molecules are far too large to pass through the pores of the nuclear membrane. Another reason it can't leave the nucleus is that these DNA molecules hold the original copy of all instructions for future generations of cells. Ribosomes, which have functioned to synthesize proteins, bind to the messenger RNA in the cytoplasm. The ribosome reads the code in the mRNA to produce a chain made of amino acids. There are over 20 different amino acids that can be linked together in various linear sequences to form polypeptide chains up to 300 amino acids in length. Transfer RNA molecules carry the amino acid to the ribosome. The mRNA is read 3 bases at a time. As each triplet or codon is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. These amino acids are joined together by peptide bonds to form the polypeptide chains which make up proteins. One or more polypeptides twist and join together to form a three-dimensional protein in cells. The arrangement of the different amino acids determines the configuration of the protein. Any change in the amino acid sequence can change the shape of the protein molecule, therefore affecting the protein's ability to carry out its required function in the cell. Proteins are a vital part of the functioning of the human body. They are involved in transport, structure, acting as enzymes, protecting the body, and more. On some occasions, a mutation can occur in the gene sequence. This is a permanent change in the genetic information causing genetic diversity. Mutations can involve one or more base pairs and can be anywhere in the gene. A point mutation is a change in one base in a single nucleotide in a gene. If the mutation occurs in a gamete or zygote, this change will affect every cell in the developing organism and will be passed on to future generations. If the point mutation occurs in an adult somatic cell, the change will not be inherited by future generations, and the effect on the person will depend on the mutation. Specifically, a base pair substitution happens when one nucleotide and its complementary partner replaces another pair of nucleotides. Here, the base adenine is replaced by guanine, and therefore the codon on the mRNA strand is changed from CAU to CAC. However, these codons code for the same amino acid, which is histidine, and therefore don't change the protein or affect its functionality. A mutation like this occurs frequently, but often has no effect on the protein and is completely functional. These mutations are known as silent mutations. Because the protein remains completely functional, there is no threat to the continuity of the species. However, if the mutation codes for different amino acids and changes the protein entirely, this is when a non-functional protein is created, threatening the continuity of a species as they face environmental pressures with unfavorable characteristics.